Hello and welcome everyone to this video podcast for the Lab for Biology 211. This will be the final video podcast for the second module. So let's take a moment and go over what we're going to focus on in this podcast. As a reminder, you will need to take the quiz for week 4. It's only 5 questions this week. And the questions will focus on what you need to know in writing your final lab report. We want to spend some time talking about the design your own experiment. Your lab TA will provide you with data for you to calculate your own chemotaxis indices. As a reminder, there's no worksheet for week 4. There are some peer editing worksheets on Blackboard for you to use if you wish to use them, but you do not need to turn those in. I also want to go over some of the expectations for the full lab report. Make sure to contact your TA if you have any questions or concerns about your lab report, the writing it or the submission of it. Here's just our final roadmap for this module. You can see that today we're going to talk about the data from your design your own experiment. This says peer editing, but as I've said a few times that this will be optional, and you should begin designing your final figure as well. Then the rest of the labs for this week are meant for you to spend time writing and editing your final lab report. Your final lab report is going to be due this Friday, June 12th. So a few quick reminders about writing your lab report. It will contain an introduction, which you've had practice writing for Module 1, methods and results sections, which you've had an opportunity to write in previous biology courses, such as Biology 111 and 112. You'll need to include 2 figures with legends. These figures will come from Lab 2.2, which you did last week, and then the second figure will be from Lab 2.3, which you'll construct today using the data your lab TA provides for you. You'll also include a discussion with references. As you write the lab report, I want you to think about how you're going to incorporate the data obtained in Lab 2.2 and 2.3 and make connections to those initial studies done with the Barkman paper. So let's talk about the execution of the experiment that you designed. Now, obviously, we can't meet in person to carry out these experiments, unfortunately, but you will use the artificial data that your TA will provide for you. Now, these data may or may not be consistent with your hypotheses. In fact, often our hypotheses are not supported by the actual results. We don't know what the results will be. That's why we do the experiments. So don't be surprised if in some of your experiments, the data don't necessarily support what you predicted. What your TA will give you is the total number of adult worms placed on the plate, and then the number of worms at each pole. Now, most of you had 2 plates, so your TA will provide you with numbers for both of those plates. You will need to use this equation down here to calculate your chemotaxis index for each plate. And then you'll use those data, these chemotaxis indices, in writing your paper and making your second figure. Peer editing, as I've said once or twice or maybe 10 times, I guess at this point, you're not required to peer edit the full lab report. I still think it's a good idea, and I would encourage you to do so. And to help you in case you do decide to do so, I've provided some peer editing worksheets on Blackboard. However, these do not need to be turned in, and this is purely optional. So, making your second figure and writing its legend. Make sure you review Macmillan Writing Manual, pages 50 to 64, and I've also included some of the relevant information from these pages at the end of this PowerPoint. Also look at your lab manual, pages 75 to 80, and also remember to focus on the rubric found in the lab manual. Today, you'll make figure 2, and that figure will be a bar graph. And in constructing that, use Excel and use the data that you obtained today. Now, last week for Lab 2.2, you also constructed a bar graph, and that was your first figure, figure 1. But what will be different about that figure and today's figure? If you want, you can hit pause and think about that, or if not, you can just hang on here, and here's the answer. There will be no error bars on the graph you construct today. Because remember, you only did this once. There's only one trial. That is n equals 1. And when that's the case, there really are no stats you can do. You're just going to present the data from this one experiment. So it won't be quite as strong as if you had replicated this numerous times. And you'll, of course, write a legend for figure 2 and follow the same rules you see over here, which are going to be similar to what you followed for figure 1 and the figures for your introduction that you did in Module 1. Writing the discussion, again, review the Macmillan Writing Manual, pages 87 to 91. And again, these page numbers for the writing manual may be different depending upon the edition you have, but it'll be pretty close. You'll also find these at the end of this PowerPoint. Also look in the lab manual, pages 75 to 80, and look at the rubrics. So here's the section on discussions, and I'm showing what you need to do to get 100% on these discussions. Your discussion must interpret each experiment, that is lab 2.2 and 2.3, using logic supported by the data. For instance, maybe the experiment that was designed for 2.3 came forth because of your results in 2.2. So for each experiment, you should be able to complete these sentences in these boxes. We set out to find blank, and we found blank, which suggests blank about how genes influence chemotaxis behavior. So this is just a guide to help you think about how the discussion should be written. The Bargman paper is also very important to include in your discussion, because that was the paper that first identified the chemotaxis indices in C. elegans, and it's the paper that you used for lab 2.2 and lab 2.3. So what's due this week? Quiz week 4 is due Thursday at 11:59 p.m. So the good news is that there are no worksheets due this week. The peer editing, of course, is optional and recommended, but you don't need to turn that in. The bad news is that there's still this full lab report due, and it's due on Friday at 11:59 p.m. I extended this to Friday just to give you an extra day to work on that, but please don't put this off until the last minute. I want you to do really well on this full lab report. Remember, it's worth 200 points. It's worth a large chunk of your lab grade. So please take the extra time you have this week to write and edit this introduction. Next week, we're going to begin working on Module 3. You're going to learn about this model organism called Saccharomyces cerevisiae. It is often called the budding yeast, baker's yeast, or brewer's yeast. And that is because it is the yeast that is used in making wine and beer and in making bread. It's also the yeast that we can use to make ethanol that can be a fuel additive in gasoline. Now, for thousands of years, people have been working with yeast to improve upon these products, particularly wine, beer, and bread. Thousands of years ago, we weren't worried about gasoline. But they've used yeast to improve either the taste, the efficiency, or the yield of these products. And one way that we currently use to investigate yeast in making these products is to try to understand the genes in yeast and how we can manipulate those genes to alter these products. And so next week, you're going to begin by designing and analyzing data from a yeast genetic screen. And down here is just a generic yeast genetic screen, similar to what you're going to do in lab, where under certain conditions, some of these cells grow. For instance, this one here is growing on this plate, but it's not growing on this plate. There's something about this gene that has been mutated, because this is the same colony here and would be here. There's something about the gene that is mutated that allows it to grow on these conditions, but not on these conditions. And that's what we're going to be looking at next week. All right, now the next several slides I'm not going to go over. These are just placed here to help you easily find the information you need to write your full lab report. That's all I have for this video podcast. If you have any questions at all, please make sure you contact your TA or contact myself, and we'll be happy to help you with any questions you might have. All right, good luck.